Assalamu alaikum. Do I am audible? Am I audible? Okay, thank you. Do I am Anna? Uh, okay, let's start today's session, the final session of life sciences research workflow. Uh, a very warm welcome to day three of our life sciences research workflow workshop. I am thrilled to have you all here today. Over the last couple of days, we have journeyed from hypothesis to experiments, and now it is time to answer a big question. How do we share our science with the world? Today is about making your voice heard, whether it is through a powerful research paper or even a poster at an international conference. Because let's face it, doing the research is only half the job. Exploring it well, that is what sets great scientists apart. So let's get started and learn how to turn data into impact. So now I am going to share the screen to all of you. Okay, thank you. Okay, it's the day three, the final day of our workshop, and in this session, we will talk about how do we share our science with the world, how to share your work with the audience, with the technical and non-technical people. So let's start. Okay, if you can't explain it simply, you do not understand it well enough. It is Albert Einstein. Okay. Welcome to the Life Science Research Workflow Workshop. It is now start from my introduction. My name is Ras. I am a BS Genetics and Molecular Biology scholar at University of Health Sciences and bring a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics and evolution, molecular diagnostics, and many more. Okay, the objectives of today's session is first of all understanding how to interpret and present the experimental results, and also learn the structure and writing style of research paper. And also we explore the simple ways to present your research in class, conferences, or journals. Okay, first of all, we start from what to do with your results. You did an experiment. Okay, now what? First of all, you organize your results clearly. Okay, the results you got, you organize it clearly. Then label the gel images. You do the gel electrophoresis. The images you got by gel doc, label them clearly. And if you got the graphs in the delta city values, that is actually in real time PCR, you got the result in delta city values. Think about what is your result showing. Okay, the, res- the result is actually you got the result, but you have to interpret that result. Does it support your hypothesis? The hypothesis you make, do it support you or raise some questions? If it says raise some questions, then revise your hypothesis. And if it supports your hypothesis, then repeat it again. And if it also reproducible, if the hypothesis is supported and the result is reproducible, it means that your research is very good. Okay, as I said you that as you see in here, it is the image of gel. Okay, the image of gel is simply like this white, black and white, okay, like that, black and white. But you have to label it like this is the marker DNA letter, DNA letter, and this is the actually the samples 40, 41, 42, 43. These are the samples, and the K positive and K negative are the controls. Okay, that are the controls, and the band size you got is actually you can write here, label here. Okay, this one is the 100. This is the 100. This is the 200. That one is the 300. That one is the 400. That one is the 500. The brighter are 500. Then 600. That DNL address is 700. That one is the 800. That one is the 900. And that one is the 1000. Okay. And the band you got in the 40. In the 40 number sample is about 158. Okay. You have to label it. Okay, then interpreting the results you got, then you have to interpret it. For example, you got a PCR gel images, like in that. The lane one is letter. Okay, the lane one that is one is letter. The second one is the sample one, and uh, the third one is the sample two. Okay, the lane one is letter. The lane two is sample A. Sorry, the sample A. and band at 500. It is about to 500. And lane 3 is sample B, no band. Okay, we got no band. So what are the interpretation of that result? It means that sample A has amplified DNA like that. Okay, but sample B may have failed amplification due to poor primer or DNA quality. The screen is shown to all of you or not? I don't know. Amna said is questioning here. No screen show. If screen is not shown, please tell me. Okay, okay, thank you. Then Amna said if the screen is not shown to you, then kindly rejoin the class. Kindly leave that class and rejoin it. Okay, okay. Now we will dive into the formatting your results. Okay, the result you go get now present data using like tables. You can make tables. You can make tables like there are the samples, the results you got, and like that the methodology you use, the interpretation, the controls, the like that. Okay, for example, the nanodrop readings of DNA concentration. The DNA concentration, like there in the second day of the workshop, we talked about the 1.8 to 2. Sorry, the 2.0. The DNA concentration is good, is perfect, have no any contamination. Okay, so you have to write it in the way like in the 40, in the 41, the 42, and the 43, and write here, for example, 1.9. Then you got 1.7, then you got 1.95, then you got 2.0. You can write here. And the graphs. You can also go to the bar charts for expression levels. Okay, as already we talk about the data city values, you got in the real time PCR. You have to show that in data that results in bar charts, and you also it formats your results in figures, labeled gel photos, the microscope images, like that. And this one is the graphs, like the bar charts, and that are the microscope images. And this is not the simple microscope, that are actually the electron microscope images. Okay. The pro tip. The pro tip is always include the legends, labels, and units. What is legend? Legends is actually the short sentence, short sentence of the table. You can also include in your gel photos in microscope images in the bar charts. Okay, if you have any questions in your mind about what to do with your results and formatting your results, if you have any questions about it, you can ask me in the chat or by using the mic. If you have any question till now, you can ask me in the chat box or by using mic. If you have not any question, kindly type no. Okay, Amna sir, you have a question. Could we work on real world project live by grouping some member of same field? Uh, yes, you can. If your background is life sciences and about the scientific research, you can do it by making groups, and by this, someone will work on like writing, someone will work on experiments or the lab work, someone will work on uh, synopsis approvals. So, like that, all people can work together and make impact in real world. Mm. Malik Shahzan, you have questions or when we will get our certificate? Yes, after completing this final day session, I will share the contacts of my collaborating company, the life sciences enterprises, that will definitely provide you the e-certificate to all of you. हाँ दला 10:30 के बजे तक आना 10 के बजे तक Can you make the group head for work as a team on publication? Amna said, if you want to work with me, with my team, you can because my team has already worked on colorectal cancer, also worked on breast cancer, like that in the scientific research. If you want to join us, you can contact me by using email or by my WhatsApp number. I will show you the WhatsApp number at the end of that session. Okay, Amna Mariam, we can use this platform for networking. Should we share our LinkedIn accounts in the chat box? Mm, it's not for that, Amna Mariam, because if you want to contact with me, can you contact with me by using email or by using my WhatsApp number? Okay, but don't make networks here. I will definitely, if you contact with me, I will add you in the WhatsApp group that are actually have background of life sciences, so you can work together. Okay, Mariam, baby, contact with me on email. I will definitely provide you the contact number of my company, License Enterprises. Actually, actually collaborating company will actually provide you the certificate. Okay, if you have any question right now about the okay, okay, contact me. Okay, now I'm going to share you the screen again, and we will definitely. Okay, my contacts will shown to you at the end of this session. I hope so. This screen is shown to all of you now. Get so we have to continue the next part of our session. Okay, basics of writing a research paper. A standard structure includes title. First of all, the title, the research, the scientific research is actually incompleted if the title is not present or the abstract is not present. Or even these eight things are not present or not well structured, it will not be perfect paper. First of all, the title, then abstract. Abstract is actually the summary of your research, then the introduction, then the materials and methods, then results, then discussion, conclusion, and references. So we will talk about one by one in the this session. Okay. First of all, the title and abstract. Okay, title should be clear and specific. It should not be very much expanded or not containing like it is very broad. It should not be very broad. It should be clear and very specific to some topic. So what should be the abstract? Abstract should be brief summary. It should not be more than 250 words. It should be between 150 to 250 words. Yeah, it should contain background. It should contain what is actually the purpose of your research. It contains the methodology of your research. It contains the key results and also the conclusion. Like this is the abstract image. First of all, the purpose. Kindly take screenshot of this slide, and you can check it later also. This is the purpose, then the method, then the results, and then at the end. The conclusion. Okay, then introduction. The introduction includes the background of the problem. What is actually the problem? What you are gonna resolve something. What is actually the problem is. Then what is the importance of the study? Is the problem is broadly acceptable or not? What is the importance of your study is, and what is the objective or the hypothesis? Okay, like in the PhDs or for example, in the masters or in the master's research, the funnel technique is used, in which the broad to generate new unanticipated insights, then you have to make it more specific to collect the details. Okay, you have a very deep knowledge about about your research. But if someone is non-technical, is not have a much knowledge about your research or about your topic, so you have to be more specific about it. You should give something to someone that they want. So you should be very specific. For example, the colorectal cancer, in which it is the for example it is the third most common cancer globally. Okay, and the APC gene is frequently mutated in such cases. This study aims to evaluate its expression in patient samples. Okay, in this paragraph, you mention the APC gene is frequently mutated in colorectal cancer. Okay, but you don't mention, you are not mentioning here the WNT pathway is actually. overexpressed, and then the C make that is actually the proliferate the cells, and the survival make the anti-apoptosis. Not like that. You should be very specific to something, so everyone uh, can read it and can understand. So now we talk about the materials and methods. It should describe the sample collection, what you collect, what is your actually the methodology of your research, the sample collection, and then you do the extraction, then you do the primer sequencing, and then you do the PCR protocols, and the equipment you use, and for example, nanodrop, thermocyclers, centrifuges, microfibers, tips like that, and everything you can write in the materials and methods. So keep it reproducible and clear. For example, you observe something, okay? You observe something and you got some problem. Then you make a hypothesis. If X is this, then Y will happen. This is the hypothesis. Then you do the experiment. Okay, you choose two groups. One is the control group. The second one is the experimental group. The patient, the or like that, and you got the results. The results you got is inconsistent with the hypothesis. Okay, if the result is inconsistent with the hypothesis, then you revise the hypothesis. But if the evidence, the results supports the hypothesis, then you should repeat it, and it should be reproducible and clear. And if you got the results again, you make the scientific theory. Okay, and then results. The results should just state the findings, what you find. It should not interpret your results. You shouldn't interpret. Yes, I got this one. Not like that. If you got that, for example, the average DNA concentration was 80 nanogram per microliter. Only like that. You are not mentioning the methodology. I just take the sample and then microfiber, then we fit the tip, the P100 tip on it, and then use the nanodrop machine, and I got that result. Not like that. You just simply state the findings. I got that result, something like that. For example, I got 52% result, like yes, a significant crisis. 10% don't know. Like this is actually the survey base. This is actually the survey based research. And then discussion and conclusion. Discuss what the result means. Okay, now you can interpret your results here. Did your results match the expectations? Are your results uh, actually supports your hypothesis? Are there limitations? Are there the limitations are present or not? You can also mention it. What is next? If you got that results, or you got that if the hypothesis is supported by your results, then what is next? Okay, and at the end, you can also conclude your research, your writing with 2 to 3 lines of the conclusion. And at the end, you can also mention the references, use proper citation style like APA, Vancouver, MLA, Chicago. The mostly used are one as the AP, and in which it is actually the Google Scholar, in which if you explore the Google Scholar and type something like that, breast cancer, like that, okay, and then click on search, you got the results. Okay, in the end of the each paper, you got the word site. Click on me. You can copy paste your that citation into your research paper, like in the MLS style or the APS style or the Chicago. Simply copy it and paste it into your research. Uh, in the NCBI, you got the citations. In the journal article, you got the citations. In the lab protocols, you got the citations. The tip of using the references is use tool like Zotero, EndNote, Mendeley, or the Google Scholar. If you have any question right now, you can ask me in the chat. हेलो हाँ जी हाँ जी है ਉਹ ਉਸ ਲਈ ਪਤਾ ਨੈਟ ਨਹੀਂ ਸਹੀ ਚੱਲਦਾ ਪਿਆ ਤੇ ਮੇਰਾ ਸੈਸ਼ਨ ਸੀ ਨਾ ਹੁਣ ਲੈਂਦਾ ਪਿਆ ਹਾਂ ਉਹ 3 ਦਿਨ ਦਾ ਹੈਗਾ ਸੀ ਅੱਜ ਵੀ ਹੈਗਾ ਤੇ ਉਹ ਬਸ 10 ਕੁ ਵਜੇ ਤੱਕ ਖਤਮ ਹੁੰਦਾ ਫਿਰ ਮੈਂ ਆਉਣਾ 10 ਸਵਾ 10 ਵਜੇ ਤੱਕ ਹੈ ਮੈਂ ਫਿਰ ਕਾਲ ਕਰਨੀ मैं करना सवा 10 बजे तक हाँ चल okay in 5 minutes this call will be end so I am going to share you the new link to all of you kindly join the new link kindly join the new link and leave this call as soon as possible because in 3 minutes this will be end. Kindly join the new link. Leave this call and join the new link. Muhammad Abdul Rahman, and an actor Anna Tarbala Shrine. Kindly join the new link in the chat box. I already pasted the new link. Kindly join this. Kindly join the new link. So welcome back to the new link. Okay, if now have an if you have any question, you can ask me in the chat box. We will definitely answer you at the end of this session. We have to continue this session because already it is very late. Amna said, I will answer this question at the end of this session. Because at the end of this session there is a exclusive question and answer section. If anyone has question, you can ask me in the chat box. Just write your question here. I will definitely answer you in the end of this session. Okay, now let's start. Let's continue the session. And I am going to share you the screen to all of you. Okay, now presenting your research. Now you write your research, you do everything. Now you have to present your research. You have a lot of options. The some options are mentioned here, like you can present your research in class. You can also make a poster and can show in the conference. You can also present your research orally, and you can also journal. Okay, okay. Some tips like that: prepare the clear slides and posters. If you prepare your slides or the posters, kindly prepare it clearly. Practice delivering your message to both technical and non-technical audience. Okay, you have to simply tell your research, your result, your discussion, your conclusion to all people from technical, non-technical areas. Tips for better presentations: be clear and concise. First of all, be clear and concise. Avoid cluttered slides. Means a lot of things are written in the slides, like and no one has to show it, and everyone feeling bored. Not like that. Use visuals, graphs, images. By using graphs or images, the visual appealing is become high, and it will not be like it is just taking some lectures. It's not equal to the lecture. Your presentation is about your struggle. It is about your 2 to 3 years. If you are doing PhD, it is about 3 to 4 years. If you are doing your work on your thesis of your master's program, it is your struggle. So make it very clear and concise so everyone can enjoy it. Practice your timing because if your timing is very is not good, then your presentation will not be perfect. You have to practice your timing, use mirror, or you can also practice in front of your friends or family. And know your audience, background and qualification, and according to your audience, make your presentation more appealing. Okay, thank you so much. Now, mini task for day 3. Kindly take screenshot of this slide. Okay, if you take screenshot, you can also take screenshot. And now, what is the agenda? Create 5 slide summary of your own real or hypothetical research project. Okay, make summary of your own. You can make it on PowerPoint. You can also use Word. Uh, what is actually included in your summary? First of all, title and abstract, material, methods, results, discussion, conclusion, references. Okay, all 8 things should be included uh, in your summary. Okay, you can submit it via WhatsApp or email. Okay, you can also uh, take screenshot of it. You can also contact me if you have any question. If you want service from me, if you are doing your research, I can also help you in your research, and we can also make group research also if I am free because I am very busy in our projects. If I am free, I'll definitely contact you. Okay, thank you for being part of this wonderful journey. For further workshop announcements and a lot more, you can contact me on LinkedIn. You can follow me. You can also send me connections. I will definitely want to connect with you because you are very enthusiastic, and I want to uh, connect with you. Okay, the recordings of the workshops will definitely uploaded on YouTube. You can also check it out. The name of my YouTube channel is Ali Taraz. You can definitely join with me at this. Okay, if you want to contact with me, you can also contact with me by email, and you can also contact with me by using WhatsApp. Okay, if you have any questions, you can ask me in the chat box. Now, this is the section of question and answers. I will definitely answer your questions in this section. If you have any question, you can ask me in the chat box. Okay, the first question is from Amna Said. The question is, please elaborate of structural difference of the review article, research papers, thesis, synopsis, and systematic review. It's a long question. Okay, I will. Okay, first of all, it is about a review article. Okay, the review article is like a summary of the existing knowledge. Okay, if something is present and a lot of papers are present, like it is about breast cancer, and almost 10,000 papers are present. No one want to read all 10,000 articles. Someone want a summary of all the articles. No, 10,000 articles, maybe 200 articles or 250 articles must be summarized somewhere. I want to I want to read that review article rather than go to each research paper and read that separate. Okay, and the structure of review article is same. Okay, it also contains the title, the abstract, and the introduction, the main body like methodology and experiment. Sorry, it contains the methodology, not the experiments like that. The methodology, the mention in the review articles you can mention in your in the main article you can in mention in the review articles. And the discussion and conclusion you can also write here. And the most important thing you can find in the review article is the list, the long list of references. Okay, you can click on any reference and you can also the reviewer, read the research article separately also. Okay, and in the review article, the experiments or the reason that is not included because in the review article, we don't have to do the experiments. And now, the next is research paper. Okay, research paper is actually the article. It is actually the review art. Oh, okay, sorry, it's the original article. And the structure is same as I shown in the slides. It is same. It contains the title, the abstract, the keyword, the introduction, the materials, the methods, the experiments you do, and the results, the conclusions, the discussions, and the references. Okay, in the research paper you can mention. And what is the thesis? The structure difference is the thesis. It is actually the comprehensive documents, okay, in the word file. It is actually about your research you have done by experiments. It is not about your research paper. You have to submit your thesis before your research article into the journal. If journal accepts your thesis, then it will publish your research into their journals. Write your thesis and submit into the journal. And structure of the thesis is same. It also contains the title. It also contains the acknowledgements, the certifications, because the thesis actually submitted into the journals. If you are doing your degree, if you are doing just job in R&D, research and development department of some company, then you don't have to submit the thesis. If you are doing your degree in like the master's degree or the PhD degree, then you have to submit the thesis. And that is why it contains the contents, abstract, introduction, the literature review you do, the methodology you have to apply, You have already applied, and the results, discussions, conclusions, references, it is all like same. And synopsis. Okay, synopsis is actually the research proposal. Okay, Amna Mariam has question about research proposal. Okay, so it is actually the initial question. Synopsis is actually the research proposal. Before you have before you have actually started your research, you have to submit your research proposal for getting grant or getting permission from your university or from your company. You have to submit the research proposal or synopsis. It is actually the brief plan of your research. It is like the timeline of your research. It is actually about the method you use. It's actually about the problem you want to solve. Introduction. It also contains the objectives. Okay, it is also about the you have to write the hypothesis, literature review, methodology, and most importantly, budget. You have to mention the budget, and at the end, nonetheless, references. And systematic review, it is actually you have to critically assess the all articles already existed about some specific topic. Okay, you critically assess it, and you make some predefined methodologies to make some hypothesis. And sorry, the internet connection is very slow. Sorry for interruption. Okay, the systematic review is actually you have to critically assess and synthesize all relevant studies on a specific question. Okay, by using like predefined methodology, and then you discuss about the results and your conclusions. And the structure of the systematic review is totally same, and it often also includes some flow diagrams and workflows, and it also include the meta-analysis, because in the meta-analysis you have to read 150 to 200 to 250 research articles, and then you make some conclusion. Hope so your question is answered. Okay, now the next question is of uh, so if I am pronounced correctly, Rathin Rathin Das. Okay, the question is, how do we decide which statistical statistical test is appropriate for a particular data set? Okay, it is actually about your experience. If you're very uh, if you are fresh in your field, then it is very hard to decide. But you can choose appropriate statistical test. Depends on several key factors. Actually, we will I will definitely guide you here. First of all, you have to determine the type of data. What is actually your data is? Is your data is categorical, like yes or no? Yes or no? It is like a qualitative data, or it contains some like numericals from 1 to 10, like 1, 2, 3, 4, 5, 7, 8, 9, 10. Then, first of all, you have to determine your type of data. Then you define your objective. You can compare your groups and looking for associations, and you can also predict the outcomes. Okay, like that. And by this, you can evaluate, you can decide the statistical data, te statistical test for a particular data set. Okay, Amna Mariam have question how to write a good research proposal. I have already mentioned in the first question. Do you have any template or sample related to life sciences? I see often during master's PhD application, a good research proposal is invited to get scholarship. Okay, Amna Mariam, yes, I have a lot of templates of research articles of the research proposals, and also have templates of thesis and like that. Because, but it is, I think so it is not legal to copy someone's research proposals, because you have to, if you are very passionate about your research, you can make it by yourself. But if you want, if you're fresher and just want to continue, just want to start your research, you can definitely use someone's template. But the objectives are actually matter. If your objectives are very clear and your problem and the problem you actually identified is very clear, you will definitely your research proposal will definitely be approved. Okay, Ratin does have question. What are some what are some sorry, where are the question? What are the some common reasons get research papers get rejected? How can we avoid them? Okay, very good question, Ratin Das. It is research papers are often rejected due to mix of scientific structure and ethical issues. Because first, the most important is ethical issues. Because if someone is not clearly defined the problem actually he defined, sorry, the question, the problem he identified. So that is why their research proposal or their actually paper is rejected. And mostly the poor writing and structure, if the structure is not very up to the mark, that is why your paper can be rejected. And inadequate literature review, you just review two papers. Okay, and by review your just two papers, you just doing your research and uh, want to publish the uh, want to publish the paper into the journal, it will definitely be rejected. You have, if you are very passionate about the research, about the topic you have decided, then you have to review minimum 50 research articles, minimum, and mostly it should be review articles. And the I think so copy someone's results and paste it into your research paper, and it contains copy paste systems. So that is why your research paper can be rejected. Also, your question is answered. Okay, so how we can avoid it? By avoiding these points, you can definitely avoid it, and your paper will definitely be submitted and will definitely be accepted. Okay, Amna Maria have question. Do we use any statistical analysis while writing a review paper? Yes, you can. because actually the meta-analysis by uh, when you review you do the literature review, you can definitely analyze this. If you are sorry, if you read almost 200 papers, and in 200 papers you concluded, if if uh, if the number, for example, the concentration of something is greater than 150, then the result will be positive, and if it will be less than 150, then the result will be negative. This is your conclusion. This is your result by almost 200 or 100 sorry, the research articles. So you can mention your results about your conclusions and your mm, methods. Okay, Priya, Saikia have question. Where can we make research posters? Like which application to use to make them? For research for posters, simply you can use Canva. If you write research posters on Canva, you can definitely have a lot of research posters, templates of research posters in Canva. You can just change it into your the what the problem or the objective you have, and you can shown into the conferences. And if you don't want to use Canva, you can also use Microsoft PowerPoint, because by Microsoft PowerPoint you can make it research posters. Uh, 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 and nowadays people choose just one slide, okay, just one big slide, and just write first of all the abstract at the bottom of sorry, at the upper left side, then introduction, then uh, about methodology, then about the discussions and conclusions and results and references in just one slide, and thus just zoom into some parts, and mm, you can read it. Okay, and okay, Rathin Das also answered the previous question. You can use Microsoft PowerPoint or like Google Slide. It is very useful to make values. Yes, Rathin Das, thank you for answering the question. It uh, it is very good, and I am happy you are answered the question. Okay, and I have question: how to find good research articles? How to best evaluate the research article and to study itself? Okay, Anna, if you want to find a good research articles, then in the first session of this life sciences research workflow workshop, I have mentioned that for good research articles, you will be very specific. You will be very specific to your research. You will be very specific to your about your the problem you identify. If you are not specific, if you are working on bacteria, okay, it is not very specific. You have to uh, work on something like E. coli or Staphylococcus, something like that. You should be very specific, and you can also use the Boolean operators in first session. If you have, Anna, you have actually joined that session. If you want to recheck that session. You can also check the recordings on my YouTube channel. By this, you will definitely be answered very well. Because in first session, I have also mentioned how to find good research articles in that section. If anyone has question right now, you can ask me in the chat box. I am very happy because everyone in my audience is active. Okay, Ratin Das, thank you so much. I am very happy to take this review. Okay, thank you so much. I will definitely send you the email to all of you. You can contact the life sciences enterprises and you can get the e-certificate of it. It will the e-certificate. I think so. It is not free. It will take almost $4 to $5, but I think so. It is very economical. You can pay it, but in my in my side, I can deliver the workshops free, but the e-certificate is company's rule, so you have to pay it for certificate. But it is valuable. I think so. It is worth it. The $4, I think so. I think so. It is just a minimum amount you can pay and get the e-certificate and also the letter of recommendation. You can use it for applying the scholarships. So thank you so much for this wonderful journey or 3 day workshop. Also, you will definitely join the next sessions of the workshops. Also, I am very uh, thankful to all of you. Allah Hafiz.